Welcome and thanks for joining us for our June Lunch and Learn. I'm glad everybody could be here. I'm Julie Brown. I'm the Raptor Migration and Programs Director at HMANA. We're excited to welcome Bryce Robinson today, who's going to be talking about the red-tailed hawk project and what he's learned about this species through his research, migration tracking. Some of you may remember that we had Nick Aliotto as a Lunch and Learn speaker actually 2 years ago today, or 2 years ago this month. And he spoke about his red-tailed hawk research in Michigan, and I think Bryce is going to touch on some of that research today. I'm going to be facilitating things today along with my husband, Phil Brown. He's the Director of Bird Conservation at the Harris Center for Conservation Education here in New Hampshire, and he's also the HMANA Tour Committee Chair. So a few things before we get started. As usual, this talk is being recorded and we are live streaming on our Facebook page. And you can view any of our Lunch and Learn programs on our Facebook page afterwards or on our website. Just scroll down to our What's New section of our website on the home page. And I think last month is not yet up, but it should be up soon, our presentation on nighthawk migration. So we have disabled audio and visual video for everybody today. Found that it's the best way to reduce distraction during talks. But even though you're muted, we always love to connect with all of you. So we encourage you to use the chat window and ask questions for Bryce. You can make comments there. Phil is going to be reading questions to Bryce at the end. And we really love hearing where you're tuning in from, maybe what Hawkwatch you represent. So feel free to add that. And if you don't already get our e-newsletter, you can write your name and email address in the chat, and I'll take note of that. And that's a great way to just stay up to date with HMANA news and events. And that usually goes out once or twice a month. So we won't inundate you with Raptor information. So with that, I'll turn things over to Phil to introduce Bryce.
Bryce Robinson is a PhD candidate at Cornell University and at the Cornell Lab of Ornithology, where his research focuses on the evolution of diversity in the red-tailed hawk. As part of his work, Bryce co-created and leads the Red-Tailed Hawk Project, a working group aimed to fill knowledge gaps in our understanding of the life history and evolution of the species. Additionally, Bryce is an ornithological illustrator whose work has been featured in scientific papers, on journal covers, and in logos for organizations and events. To see Bryce's work, visit www.ornithology.com. That's ornithology with an I. Julie's putting it in the chat. And to learn more about the Red-Tailed Hawk Project, visit www.redtailedhawkproject.org. And I'm happy to turn it over to Bryce.
Excellent, thank you. Thank you all for inviting me to talk and for joining in to focus on the red-tailed hawk. I'm today going to present the Red-Tailed Hawk Project, which is this collaborative effort to conduct research on this species. To start, I want to give an overview on the species. I like to start the talks by showing just this picture of a red-tailed hawk, because I think that everybody who's tuned in here today probably is quite familiar with this species, and because of that might not consider it to be one of the most interesting species that we have, at least here in North America. I am of that mind, and so hopefully today I can walk you through why I think that, and you might gain a stronger appreciation for this common but really amazing bird. Many of you probably already know this, but this slide is for those of you that don't. The red-tailed hawk is comprised of many different subspecies. It's what we call polytypic. There have been 16 described subspecies. A portion of these are recognized by most authorities today. But for the red-tailed hawk project, we see no problem with recognizing all of them, because I think there's a lot to learn about these different. Generally speaking, this diversity is mostly, the descriptions of this diversity is mostly based on differences in plumage across their distribution. The other important thing to understand is that this difference in plumage diversity, or this plumage diversity, generally correlates with differences in the habitat that this species occurs in across its distribution. Here's another really fun photo that I like to share to really drive home how much plumage diversity there is in the species. This, of course, obviously is only in tail pattern and coloration, but you can see here how much differences there are within this one species. And that's in a lot of ways very exciting and very perplexing. Another really interesting thing about this species is what we call polymorphic in plumage and a portion of its distribution. This just means, for those of you that aren't familiar with this term, different color types. And generally those color types are a dark and a light morph like what you see here, although there is somewhat continuous variation in that as well. This plumage polymorphism only occurs, or is thought to only occur, in a portion of the species distribution that's outlined here in the brown. So you can see that it's generally restricted to the western portion of the species distribution, and subspecies like borealis and in the Caribbean, they're considered monomorphic. They don't have any dark morphs at all. Another interesting pattern that's found in the subspecies that have plumage polymorphism is that in the extreme northwest, in the subspecies Harnelii, most of the individuals are actually dark. If you move south into the west, like in the distribution of Coelurus, it flips. Most of the individuals are light. So this causes this really cool pattern, makes one wonder, why is there such a switch in the proportion of dark? And that's something that I think is a really cool question that we can try and answer here in the coming years. Another point of an important point of context for understanding this species at present is that shown here in the eBird status and trends map for the breeding season, this species is among many other species here in North America that is showing declines across its distribution. So although it is common and generally considered a species of least concern in its IUCN status, it is showing declines. So there's an argument to be made here, I think, about focusing on this species to make sure that we can keep this one common bird common among other common birds as well. Now I'd just like to transition into focusing on a description of our mission as a working group, the Red-tailed Hawk Project. It can be simplified in the one sentence that we aim to conduct that research broadly. And by broadly, I mean what I'm doing here today. I'm not just going myself to publish papers that are for other researchers, but also I and the other members of the group are hoping to communicate the findings of that research to people that are just generally interested. And that might be people who just learned about what a red-tailed hawk is, maybe a day before they talk to us or find our website. I like to share this photo because I think it captures the spirit of our working group really well. This photo's, my friend Greg took this photo in southwestern Idaho. I was out catching red tails there during the breeding season to get blood samples for some phylogenetic work. And I stopped to process this bird in this farm, in this really rural area, but it was pretty near to this house. Came out to see what I was doing. They saw me holding this hawk, and they didn't know what a red-tailed hawk was. They knew what hawks were, but I got to give them a description of what I was up to and why, and some cool things about this particular bird that I think really, I hope really brightened their day, gave them an appreciation for the birds that they see, and really maybe sparked an interest in the natural world at least. I I also want to give an overview of our project website, because I think what we're trying to do with this website is provide resources for anyone who's interested in learning more about the species. So I'll just touch on a few of the things that we have in our website. If you haven't gone there, please visit. First, and perhaps one of the most important parts of our website is the blog. Here we share updates on things that we're finding out about the birds that we have tagged. I might be sharing some cool things I'm finding out as I go along my research in the genomic realm or the evolutionary realm. But this is kind of like a newsreel for our project. The blog here also mirrors what we put on our Facebook page. So if you're more into monitoring news on Facebook, you can find our stuff there. But this is just a description of what we're up to. So you can tune into that every once in a while. We also have this research tab that has different pages that describe the focuses of our working group. That includes the movement ecology, the taxonomic work and genomic work that I'm involved in. And then there also is a page that is an overview of all the researchers who are participating in the project, so you can get an idea of who are the faces behind all this work that we're doing. On the movement ecology page, I'm actually going to re-engineer this page, but at the present, we have this movement ecology page that is an overview of many of the individuals that we have tagged. So you can go there and see the phenotypes for all the individuals, and for cases where we have full cycle movement for that individual, we've included a map that shows where that bird winters, its spring migratory route, where it breeds, and then its fall migratory route back to the breeding grounds. We also have this subspecies tab, and this is something that's definitely under construction, but at the top of the page, we have that map that I showed in the beginning of the talk that shows where each of the different subspecies occurs. And then below that, we have a list of all the subspecies, and eventually we'll have a fully developed identification to subspecies from one another. So you can be armed with all the features that you might use when you see your hawk in the field and separate one from the other. Also you can see here at the bottom of the page, there is a link to looking at Borealis on the Macaulay Library. So you can actually, for the subspecies that it is relevant, you can link through after you've looked at the illustrations and tried to learn some of the plumage features that we outline in these figures. You can actually go to some photos and see if you can see those features on birds that are in the Macaulay Library. So now I want to go a little bit more in detail about our mission briefly, because to conduct research and communicate it broadly, that's a little bit of a 30,000-foot view. But I think that mission statement can be broken up into a few different areas. And the first, really want to figure out, well, first promote a collaboration, but figure out how to make collaboration between many institutions and many researchers easier, more effective, and more impactful. As a working group now, we have many organizations that have researchers that are participating in one way or another by providing resources or data. As you can imagine, each of these institutions also has their own sub-network. So in this way, we're hoping to broaden our network so that we can have a flow of information about what we're all learning about red-tailed hawks, and also data sharing so that we can empower larger data sets for our research, something I'll touch upon here in a bit. The real objective of these collaborations are to fill knowledge gaps in the species. It might surprise some of you to know that the red-tailed hawk, even though it's this common species that's distributed on the continent in Central America and the Caribbean as well, there's still a lot of fundamental things we don't know about the species ecology, its life history, and its evolution. And I just put this word cloud here to kind of hit that point home. There's still a lot of work to be done. And so we as a working group are targeting those areas to help fill those knowledge gaps, and really by filling those knowledge gaps, we aim to create a model, something of a model system from this species that can be applied to raptors in general, or maybe birds in general. Some of you may be familiar with the idea of what a model system is. Rats or mice, lab mice, or zebrafinch for birds. But for this species, it's a wild species. We're not, at the moment keeping in captivity. What exactly do I mean when I say a model species in relation to the red-tailed hawk? Because this species is common on landscape and relative to other raptors, easy to capture and work with, we can use it to test, or we can conduct research on this species that might give us ecological and evolutionary insights that can be broadly applicable to raptors in general. So we can learn some things about the evolution and ecology of raptors as a whole, or even taken further, like I said, to birds and even in vertebrates. But because, again, it's common and widespread, easy to capture, we can use this species to test methods and protocols that, we might have the room to make errors that we will have less room when we're working with more imperiled species, or species that are harder to access. So we can learn the hard lessons on the red tail and apply them to other species. And then again, we can use this species and the red-tailed hawk project to push forward ways to conduct effective collaboration efforts and bring raptor biology into the big data realm, and empower future research. What I'd also like to do, or what I'd also like to see from the Red-tailed Hawk project, is really to feed all of the information we're getting to strengthen the Birds of the World account and really develop this robust and comprehensive overview of what we do know about the species. And so we as a working group are trying to do that. Now that I've shown you what we're trying to do, I want to transition into our approach and how we're going about doing it. And that really is founded, or it centers on the idea of the extended specimen. Many of you, if you've hung out around museums, you might already know this word, the extended specimen. It generally comes from vouchered specimens, where you have a round skin and a number of different things that are attached to it. Information on when it was collected, whether or not it was a male or a female, any data that's collected to that individual, its mass and such things. The extended idea comes from this idea of taking as much information as you can when you get a bird in the hand. And we are trying to do that as a working group, both with voucher specimens, but also using standardized photographs as that anchor point for all the data we collect in the field. So when we catch an individual, we take these standardized set of photos against a neutral backboard along with the color standard. We take them in a standardized way, so we're all taking the same photos, so that we can attach all the data we gather and build these data sets that we can all use as a working group. Here's an example of all the photos we take. If you've been in Macaulay on the website, these probably are not new for you, but we take a wing out, front and back of every individual of both wings, so that the idea is that we're capturing comprehensively the plumage of this individual. So we're getting all the plumage features of this individual. But you can imagine we're also getting other information like molt from these photos. So we can have records of that data as well. All of the information that's attached to these individuals that are anchored on these standardized photographs can be added, if they're taken in a standardized way among everybody who's participating in our group, to create large data sets on molt, on morphometrics, on movement for individuals that we're putting transmitters on, breeding locations, blood samples, which can give us a number of different types of information connected together, so we can relate variation in one with another and really increase our understanding of what drives variation in some of these things in the species. So these data sets that we're building are in theory going to be large and robust so that we as a working group can share them among each other and answer any questions of anybody conducting research in our group. But really what we're trying to do is make data sets that will have a utility far into the future, so that anybody in the future that has a question about red-tailed hawk that hasn't been answered yet can leverage our data sets that we're collecting now. We're trying to think forward that way. So now that I've given this overview of the project, what we're doing, and then why we're doing it, I want to touch on some of the results that we have from some of the work we're doing. And first, I want to touch on the movement ecology data set. This is HMANA, so I think that many of the people that are, if not every person that's tuned in here, really on migration. I'm going to focus first on Nick Aliotto, who is in Michigan at Michigan State. He's a PhD student there. Nick is doing some really cool work on movement ecology in the Great Lakes region with red tails. Nick really is kind of the engine behind a lot of the movement work that we're doing as a group. As a group, we're putting out transmitters that are called GSM transmitters. And if you're not familiar with that, the way these function is that they are a GPS transmitter that takes GPS fixes on a scheduled time. So we generally tell them to take GPS points every 3 hours, and then try and communicate those GPS points back to us once a day. The communication of those GPS points is through the cellular network. So anytime that you have service out, transmitters should have service as well. You can imagine that, that'll vary across the landscape. A lot of the species that we're tagging, or a lot of the individuals that we're tagging, are migratory, wintering in the central part of the US during winter. They'll migrate north into the boreal of Canada and Alaska, and there's very poor cell coverage, if none at all, up there. So we don't hear from them all summer, and they log their data. And when they come back south during fall migration, they offload all the summer's data. So you can imagine that, October 1st time, when we're all hyped up over excellent fall migration, we at the Red-tailed Hawk Project are even more hyped up because, we're a week out from, in many cases, getting a lot of really fun insights into where individuals have spent the summer. Nick has been focused, like I said, in Michigan, he has been trapping with the Mackinac Straits Raptor Watch in Mackinac City on the northern tip of the Michigan's Lower Peninsula. Here, well, first I have to explain, for those of you that don't know this, I almost guarantee that most of you do because you're into raptor migration, but red tails are among species that don't like to cross waters. So there are many spots around the Great Lakes region that are really fun to sit and watch the spring or fall migration, because the lake edges funnel raptors and push them along in predictable ways, so we can go to places like Mackinac City and see a lot of raptors at one time. Nick is there during spring migration. There's a great raptor migration there during spring, particularly for red tails. Nick leverages that to get to trap a lot of red tails. It's been a lot of fun being up there with him. At this spot, there's a 5-mile stretch of water, and the red tails don't like to cross this until the conditions are right for them to do so. Nick's really interested in what factors are involved with that decision to cross. From the birds we tagged, we have this individual that's a great example of what these birds do when they hit this 5-mile stretch. So you'll see this bird just kind of mill about. It'll wander around here, and just imagine there's a couple more hundred birds doing this at the same time. So it's really fun to be at this point. It's to watch, but also as we're trapping the red tails. At some point, something changes and the bird decides to shoot across the water and make the jump to the northern peninsula and continue its migration north into boreal Canada. So Nick is really interested in understanding what influences this crossing behavior. And I'm sure you'll hear from him at some point down the line when he has more results and everything, so I won't steal any of his thunder. Now I'll focus on some of the broader things that we're working on. Here's a good individual that represents some of the more descriptive stuff we're getting from individuals that hasn't really been done on a large scale in the past that we're excited about putting together now. This is a bird that Jesse Watson at Hawkwatch International tagged a few winters ago. It's a dark morph harlani that wintered in the Salt Lake area. You can see here that its spring migration north went along the Front Range in Canada and then dipped into south central Alaska where it bred, and then returned down a similar route during fall migration. This is really cool to see for harlani. It's really cool because we see this really nice fidelity between the spring and the winter routes. But there's a lot of questions that this raises just from this simple full cycle track that we can answer or start to gain insights on by tagging a bunch of different individuals. Perpraki tagged in northern Idaho a few winters back. And both of these are harlani, but they have very different migration tracks, as you can see. One ended up breeding in central British Columbia. It has really tight fidelity between spring and summer, whereas the other one ended up breeding in far western Alaska. It took its spring migration north through central BC, winding through the mountain valleys there, and then on the way back south went along the Front Range and wintered in the same spot. So that's fun. What drives these differences? Is it individual based? Are there factors that are involved? Do they vary by season? So some of these individuals that we tag and track for multiple years, are they following the same route spring and summer every single year? Or does it vary, and what's behind that variation? These are questions that we're hoping to answer over the next few years, and hopefully in due time. Another thing that we're focused on in our efforts to track individuals is understanding the distribution of plumage polymorphism, something that I think many of you who are into redtails and migration, especially in the Great Lakes region, also have a lot of curiosity about. This question, or understanding the distribution of polymorphism, was actually the reason why the Red-tailed Hawk project was started. And so I'm excited to share some results on that. When we first came together, there are folks, Mark Robbins and Luke DeSico at University of Kansas, were talking with me about understanding where the dark morphs that are non-harlani in appearance, where do they come from? All these individuals that are this phenotype that are wintering in the Great Plains, particularly in eastern Kansas, where do they originate? The question is, do they breed directly north in like Manitoba, in places where we didn't understand that dark birds occurred during the breeding season, or are they originating further west in areas where we do know that that dark morphs occur, and perhaps these should be called calurus. That was the start of the project, and so we set out to catch individuals to answer this question. Actually, HMANA supported one of the transmitters that got this all kicked off. So HMANA was a part of the really getting the ball rolling for the Red-tailed Hawk project so we could get some momentum to carry our work forward. This is what we found out from the few years that we made this effort to trap dark individuals that were non-harlani in the Great Plains. Generally, all the birds, they took this really nice northwest trajectory up into areas where we already understood that dark morphs occurred. Some of the surprising things were that these birds occurred areas there in the center, ended up in Alaska, where we know is like smack dab in the middle of the distribution of harlani. But other areas like northern British Columbia, we also know harlani are there as well. So this begs the question of whether or not these represent, well, it just begs the question what population these really belong to. If we're thinking in subspecies concepts, is there a subspecies label we can actually use for these individuals? That's something that we're going to focus on in the future. But this really doesn't answer a question that a lot of the folks that are focused on red-tailed hawk polymorphism in the Great Lakes region want to answer, and that's the birds that are very few in number, but winter in the northwest or northeastern part of the U.S. where do they originate? Now that we know that birds in the Great Plains originate far in the Northwest, do these birds in the East originate from the same place? Here's an eBird map of the tag, which generally is a proxy for those dark types that occur in the East during winter. And you can see that they're spattered around. There's not a lot of them, but they are spattered around. Now, if I showed you Colurus alacensis as well, that you'd see more individuals here, but it's just, again, not a lot. But they are annual, and everybody who watches fall migration in this area knows that they see about 1 to 2 every year. So again, the question is, are these just the extreme eastern extent of this same migratory pathway that we see in the Great Plains bird? And so we set out to try and tag some of these individuals, and our collaborator, Brian Sullivan, here at the Cornell Lab, he called these individuals unicorns because catching red tails is easy if you have a lot of red tails to choose from, but when you want a particular individual, it is very difficult and takes a lot of time and effort. And I'm happy to report, if you haven't heard already, that collaboration with folks at Hawk Ridge, Ali Pisano and Frank Nicoletti, with Nick Aliotto, and all of us at the Red-tailed Hawk Project, we came together and we were able to catch these 4 individuals and put transmitters on them and get migration data from them. We were extremely excited to be able to do this. And the picture that came out from this work is that these individuals all spent the summer in the eastern parts of Canada. All these light circles that you see here are individuals are light Abieticola that we had tagged, both here in New York by myself and Nick in Michigan. So you can see here that these dark types are spending their summer within the distribution of Abieticola. It's really exciting to know that these dark types in the East, at least a subset of them, come from directly north of us and in Eastern Canada. But we wanted to try and put together a picture to really understand whether or not they actually did breed, because that's important to understand what we might call these dark types. Before I go into the data that supports whether or not these breed, I just want to give you an overview of the figures I'm going to show. Many of you probably know the terminology that we use to indicate the age of these individuals, but this number and letter here in the bottom right of the photo, this is the bird's age. This indicates that's the second cycle, or it's in its second year of life. You can see those shorter secondaries there in its wing. Those are retained juvenile feathers. So this bird's only in its second year of life, and it's a male. This dark line here, this means that the bird spent 95% of its time during what we binned as an 80-day time period, spent 95% of its time within that circle there. Color indicate areas of high use. And then this number is the amount of area that the bird spent 95% of its time moving around in during this 80-day period. And you can see this is 9,000, almost 10,000 square kilometers. That's huge, but that's what we'd expect perhaps from a young individual that's not breeding. It hasn't recruited into the breeding population yet. So now I'll show you this dark type that Ali and Frank tagged in comparison to a light morph that Nick tagged. And you can see that both are males, both are second cycles. The light morph has almost 100,000 square kilometer breeding range, but the point is that they have massive breeding ranges. These things are not breeding during the summer. These individuals just wandered around all summer, which is what we'd expect. The other individuals that we had tagged were older. They are after third cycle, or at least in their fourth cycle or their fourth year of life. And you can see that the males had about 20 square kilometer home ranges that are kind of centrally focused. This, is suggestive that they're holding a breeding territory. When you look at the females for that same 80-day bin, you can see that they are tiny home ranges for the same age class, about 2 square kilometers. That means these females are incubating and holding tight to that nest. They're incubating and brooding those young. So we think that this really indicates that these because there's no difference between the light morph and the dark morphs, and that these are predictable patterns from breeders, we think that this supports the fact that these dark morphs actually did breed in Eastern Canada during the summer. So that's very exciting. And what that means is that it extends the distribution of our understanding of the distribution of polymorphism in the species about 1,500 kilometers into the east, Canadian boreal. This is really fundamental if we're ever going to understand why we see the patterns in polymorphism that we do in the species, why some species have it, why some species don't, why there's more in some areas and less in other areas. Knowing that that there are dark morphs breeding in the east helps us tease apart what factors are involved in that, and we're excited to hopefully do that work here in the future. Another really neat migration story I wanted to tell is this individual that Neil caught in northern Idaho. It's a second cycle bird. So again, knowing what we saw in the other ones, we kind of expect this bird not to breed once it goes north, which in winter one, he caught it and it went north onto the Kenai Peninsula in Alaska. It actually ended up summering in an area just outside of Homer, which was really fun to see. The other fun thing is that it island hopped on its way north across Southeast Alaska and coastal. People that live in that area talk about there's a raptor migration here, there's a good red-tailed migration, but nobody's ever documented with tracking data. And we were excited to see this bird go through that. In the fall, it came back on a totally different trajectory around the Front Range, ended up spending the winter in Montana, which was really fun to see it spending the winter in a totally different place from the first winter where we had it. The next spring, it went on a totally different route up through the valleys of British Columbia, and then ended up back on the Kenai Peninsula. The really fun thing, as you can see here with winter 3, is that in its third winter, which was this last winter, it spent the winter on the Kenai Peninsula and decided to hang out there all winter. So it did not migrate south after that. And that is a really perplexing pattern that I'm very excited that we were able to capture. And just goes to show why just putting a tag on a bird can be excited to keep doing that and hopefully find more cases of these types of patterns. This might be common in the early part of an individual's life. Generally, when people tag birds, they want to avoid younger birds because of how many die in the first year of life, particularly. We, because of our initial objectives as a working group, we were trying to get older birds. So we made this rule where we're not going to tag anything unless it's after third cycle for a while. But eventually we started tagging second cycles. And from that, we got this really insightful thing. Nick Alioto, though, this last season has tagged a bunch of first and second cycle birds. So we're starting to gather a big data set of these young birds to see, hopefully, if this pattern might be something that young birds do often that we didn't recognize. So I'm excited for those results from Nick. Now I go through what I'm focused on for my dissertation. I'm not focused on movement data, particularly for the work I'm doing for my PhD. I'm focused more on understanding the diversity and plumage that we see throughout the species distribution. I am, love nothing more than watching raptor migration and counting raptor migration, and particularly identifying birds in flight. And that's why I was so interested in teasing apart all the differences we see between the populations in red tails. For those of you that don't know what genomics are, I want to give a brief explanation because it's important for you to understand what I mean when I say evolutionary genomics and why it's a step forward in our understanding. Genetics, you can think of genetics, we all know what genetics are or what they refer to, but as a review, genetics refer to a variation on one gene or a collection of small genes that is just a snapshot or a portion of a genome that can be a billion base pairs in length. Genomics, in contrast, is a snapshot of many different regions across the entire genome. So it's sampling regions across the entire genome to try and gather a genome-wide perspective on genomic diversity. So it's kind of, I've heard people say it's the big data of genetics. Phylogenomics, which I'll show you what the work I'm focused on using phylogenomics. Phylogenomics is a tool that we use to understand the relationships between a set of individuals and understand the evolutionary, or try and put together the evolutionary history of that set of individuals. So I'm trying to do that for all the different subspecies in the red-tailed hawk. What I'm trying to understand is whether or not plumage is a good indicator of relatedness. We see some individual or some subspecies that are fairly similar. They don't differ all that much. That's why some of this is a really fun ID challenge, right? But then we see individuals like the Harlan's hawk or the birds in Costa Rica that are extremely different from the other red tails. Is that because they are more or less related to all the others or not? So that's one thing that we're looking at. I think many of the red-tailed nuts that are watching this right now are familiar with the contentious question of whether or not Harlanii should be considered its own species. That is something that we will, through this work, gain a very strong understanding of. There's already been work by Josh Hull's lab over 10 years ago that hinted at the fact that they are they're very related to all the other red tails he looked at. Genetically should be considered a red-tailed hawk, but we will be showing you data to help support that. The other thing we're including is the rufous-tailed hawk. And I think that this is a fun one for everybody here who's interested in migration to pay attention to. I already have preliminary data, so I kind of understand already what is going on with this species or what could be going on with this species. I'm not going to spoil that yet, so stay tuned. But I will say that this Rising et al. Paper, there was a phylogeny on all the Boudionine hawks, hints at what we also found, and that is that borealis, the subspecies borealis, is less similar to Calurus than this species, what's currently considered a species, rufous-tailed hawk, is to Calurus. So you can see there in the phylogeny that Calurus and ventralis are clustering together. There might be something interesting there that's related to migration. So stay tuned for that. The other cool thing about understanding diversity in this system is how have islands shaped diversity? We know in evolutionary theory that islands have caused a lot of speciation events because a lot of these things that might occur on an island, like the red-tailed hawk that don't like to fly over water, they get isolated over time, and just by being isolated, they slowly change and shape into different species. We know that's a strong generator of speciation. Is that something that's happening in the red-tailed hawks? We might expect that these island subspecies are the most different among all of these. Is that the case? That's something that we're hoping to put together. And to do this work, some people might ask, this is such a cool species that has so much diversity. People are doing this type of work all the time. Why has nobody done this with the red-tailed hawk? And I think the answer that to do this work, you need to have good samples. So sampling the blood is the best way to do it, or getting tissue samples from recently collected individuals. Gathering blood samples from this species that occurs across political lines, many political lines, is logistically really tough, not to mention the field work that would be involved in places like Costa Rica or the Boreal of Canada to get these samples. I think it's just been kind of insurmountable as a logistical operation. And on the other hand, people leverage museum specimens, but people don't like to collect raptors. And there's a poor representation in general of raptors in museum collections, and the red tail is no different in that regard. So there really hasn't been a solid sample set for these individuals. Techniques are evolving, and people are now figuring out ways to isolate genomic data from very old specimens. And so we made an attempt to do this. We got 42 toepads from specimens in museums that are listed here. This work that I'm going to show you would not be possible without these museums giving us access to these. And so I want to just say how important that is and how thankful we are that they were willing to work with us. Some of these individuals that we sampled from these museums were over 100 years old. And this is risky to do a lot of very expensive genomic work to try and put this together. So what we decided to do was run a pilot run to see if this would work. And we chose some populations that I had blood samples for, and we also used some topaz samples, and I'll just show you the results from those. And what we put together was a perspective first that get from those over 100-year-old samples worked, which is very exciting. So we got good genome data from those very old specimens. And the other thing to take home from this is that they clustered as they should with the blood samples. So they matched. There was no false grouping based on whether it was a topad or a blood sample. So that's good. That means we can push forward with the full data set, something that I'm working on now. The other cool thing that we are showing here from these results is that the relationship between these four groups. And it really solidifies that Harlan-I genomically is very similar to the other red-tailed hawks. It's kind of nested here within this group. So as far as we can tell at the moment, well, and now that I have some insight going forward, Harlan-I is very much a red-tailed hawk, and something happened at some point very different, even though genomically they aren't that different. And we'll be following up on that to figure out if we can have an explanation for that. I'd like to wrap up by showing a few of the other little side projects, I guess, of the group that aren't our main focus, but things that have come about just from the work we're doing. And one of those is strengthening our understanding of plumage aberrations in the species. To me, it seems like in the red-tailed hawk, there might be more plumage aberrations than other bootios in general, but even other raptors or species or bird species as a whole. There seems to be a lot of birds that have plumage aberrations. Right now, I'm working with an undergrad here at Cornell to try and put together a perspective on how common this is in this species, what types are involved, or at least generally, and then gain an insight on how they impact these birds. Are they actually detrimental to these birds? Is it bad to be an all-white bird? Many of you may, when we shared this back in February, but we took this trip to Oklahoma to put some tags out. I took along some undergrads and my collaborator, Nicole Richardson. And we somehow ended up catching this really beautiful all-white red-tailed hawk. And the decision was made to put a transmitter on it. We can kind of gain an insight on what subspecies this might be by morphometrics, but we really didn't know. So we really didn't know if we were going to get a nice migration track of it going somewhere north, or whether or not it was a resident that breeds in Oklahoma. Some really cool things about this bird is that although it appears all white, it does have a dark iris, so it's not an albino. And it also has some flecking of color on its cheek and on its nape. And you can see there in the tail, that is actually color, that's not stain. So the rate actually colored. So there's something really cool going on here. And so we took blood samples, we took some samples to hopefully at some point figure out what that is. Another really neat thing is that it impacted the color of the talons as well. So why the talons and not the beak? I don't know. We ended up, like I said, putting a transmitter on. Here's a Cornell undergrad L holding this individual right before it was released. And you can see that dark flecking in the nape. After we shared this individual on social media, I got contacted by somebody who lives in that area, and he told me that that individual is actually a bird he's been watching for years and that it breeds there. I wasn't sad about the fact that we weren't going to get any migration tracks from this bird because home range, breeding home range data has for us as a working group, so it was definitely not a loss at all. The other cool thing is that we can now have a nice we could track this bird and go check on it often, even though this individual knows where it is anyway, and he checks on it often. He actually has been watching it for 7 years, and he told me that he's been photographing it for a portion of these years. And this is a photograph from a few years back. And the really neat thing compared to the bird that we caught is that it has a bunch of color in a bunch of different areas across its body that weren't shown on the bird that we captured. Now we know, after the bird's been in this area for a while, that it is breeding there. It definitely is this bird. We know that this individual's plumage has changed over time, which is something that's very cool to document and really gives us more insight into what type of plumage aberration might be going on here. We've been lucky enough to capture a few individuals with these plumage aberrations. This one that Neil caught in Wisconsin. And so we're gaining a subset of our own individuals that we have blood samples on, that we're going to start trying to put together some answers about plumage polymorphism in this species. Hybridization is something that I've always been interested in. I think as identification people, we also are probably all interested in it because we want to know how to identify hybrids. How do we know that when something's a hybrid and not just a variant of a certain species? That game can be very fun. Red-tailed hawks, we already know they hybridize with a few other bootios, like the red-shouldered hawk, the frugivorous hawk, and the rough-legged hawk. So we want to try and gather a comprehensive perspective on how many hybridization events with what species have occurred and how often they might occur. The rough-legged hawk, I think, is a really interesting one in particular because since eBird connected and people started submitting their photos through eBird, we've gained a better perspective on how often rough-legged hawk and red-tailed hawk hybrids are found. And it's actually more common than I thought, and it's really fun to see. As a working group, we've been very lucky to also capture a few individuals. This individual, Luke DeSico, and I caught in Kansas. It's a first cycle hybrid. You can see there's a almost perfect mixture of plumage traits between a Harlan's and a rough-legged hawk. And we have a blood sample from this individual. Rob Dominich also in Montana has caught 2 adults that we have blood samples as well. So hopefully over time we get a few more blood samples and we can start to understand a little bit more about hybrids, whether plumage features indicate parentage. So is some tail but has a few things off? Is that like a hybridization event that happened back in time and it's since its parents and grandparents bred with red tails back in? Or is a male and a female different from an first generation hybrid? Those types of things we can start to piece together with the blood samples. And then another thing I'd like to eventually put together is how hybridization impacts each species. Is it an evolutionary dead end? Are these individuals that hybridize first, are those parents less fit than others? Are the offspring less fit? Those types of things. So hopefully over time we'll understand that. And I'd just like to end again on Jerry's photo. Hopefully I've shown you all why I think the red-tailed hawk is such an incredible species and how much we still have to learn about it and how excited I am to be working with the amazing people that I'm working with going forward. Hopefully we continue to send out all the information that we're learning and keep you all updated if you're interested. Please reach out to me if you have any questions or want to share any information. You should always feel free to do so. Again, here's a QR code for our website. It's pretty simple, redtailedhawkproject.org. Please visit and check in often on our blog and our Facebook page. Again, it's just a newsreel that mirrors what we put on the blog. All of this work is totally a community effort. We've gotten funding from many individuals. We're communicating with individuals all the time. So there's people there's faces on the websites, there's names on the websites, but that is the tip of the iceberg of the people that are involved in empowering this work. And I also just want to acknowledge all the generous support from organizations, including HMANA, who gave us the seed money that really got us momentum to get this work going and produce what you've seen today. So thank you to HMANA in particular. And with that, I would be very happy. Time.
Okay, thank you so much, Bryce. That was fascinating work. And I'm particularly impressed by the diversity of different research techniques that you've used to better understand this very diverse species. And as Joan very perfectly put it here at the end, commenting that she's so glad to see folks finally conducting serious studies on this common species, which is the common species are often overlooked. So we do have a few questions coming from those on the Zoom. I'll read those off here. Just a few questions. You covered a lot of ground here. So we'll start with one from Ajit. The Harlani race that we see here in Colorado on our winter raptor surveys are almost all light morphs compared to 85% dark morphs in Alaska. And, I don't know, you addressed this a little bit, but is there any discrepancy that's understood regarding that you can explain?
Yeah, absolutely. There is a general trend in the increase of light morphs as you move east in the distribution of that subspecies. So in places like the Yukon and Alberta, there are more light morphs that fit the label Harlani than in Alaska itself. I think we have that general pattern described, although I think more work could be done to figure that out exactly. But it's really cool to hear about from winter raptor surveys that there might be a winter distributional trend as well. I wasn't aware of that. I'd like to hear more about that and hear about kind of the winter survey protocols that are used, because if that's fascinating, there probably are some factors that might be involved in that trend that are also involved in why we see no dark morphs in borealis, for instance, while we do see so many in Harlani. So yeah, that's a great question. I'm happy to hear about that.
Okay, thank you. Joan asks, do the migratory routes earlier on that you pictured represent one or more cycles? I don't know if
Yeah,
I know you showed several maps, but generally
so
one cycle.
Yep, those are all one cycle, and they're all the first cycle that first migration cycle that where we had that individual tagged. So I have not gone through and developed maps for multi-years for all these individuals, which is something that I or someone should and can do at some point, and will do. I think when we start to involved in interannual variation and routing and stuff and timing as well. But there are, yeah, I can't say off the top of my head how many individuals we have with multi-year tracks, but there are a bunch. They're not everything we've tagged, unfortunately, has given us more than one cycle. So yeah.
It was really interesting to see the one raptor that you showed that had the three migratory routes, or I guess the two migratory routes between Alaska and can't remember if it was Saskatchewan or Idaho.
Yeah, and something I'm very excited about with that individual and many individuals, particularly all of these birds that Nick has tagged in their first and second cycle this season, is no one really has any solid understanding of when. We have an idea and an understanding from a bunch of data, like when do these birds, at what age do they start breeding? All these individuals that we have tagged that give us multiple, could have a pretty good picture on when, at what age they recruit into the breeding population. This individual that wintered in Homer this year, I am very excited to see if it holds a breeding territory and appears to be breeding, because that might give us some insight on why it decided to stay the winter up there.
Okay. A question came in from Jess that I'll paraphrase a little bit. Jess commented that he read that Abiticola translates to living in or inhabiting fur,
Mhm.
And you wondered if there was a connection there with the name reference to the nesting ecology of that particular subspecies.
Yeah, of the furs. Yep, that's exactly right.
Okay. Great, thanks. I was curious. I know you might not have as much of a sample size, but is there a strong site fidelity in returning to breeding areas for many of the populations you looked at?
Yeah, that's something that I can only answer anecdotally from the individuals that we've tagged that I've looked at. But and we will be doing that over at some point. Again, another one of these questions that we can simply answer from the data set we've already gathered, but just haven't done yet. Red-tailed hawks in general are very they're creatures of habit in most cases. I mean, I've shown you one example where that wasn't the case with that bird that wintered in Homer this year. But generally speaking, for adults, they seem to be following the same pattern every single year. I mean, you have people who anecdotally talk about this bird that's wintered on the same power pole every single winter, and they watch it for 10 years or however long, you know. Breeding season seems to be the same where they're migratory.
Okay, I think we have time for just one more question. We got a couple more in. Just a quick question about range maps in, say, raptor field guides. I realize your data are probably informing some of the older guides a little bit. So, what can you say about how the range maps depict what you have found?
Yeah, I mean, as with science in general, that's what we're doing here. This is an exercise to improve our understanding. So I think that there's plenty of work to be done to improve the range distribution, descriptions of range or distributions of birds, particularly for the red-tailed hawk. I mean, if you compare the birds of the world or what was birds of North America, red-tailed hawk species level distribution, the eBird status and trends map or the distribution map generated from eBird data, you will see differences. And that's because we didn't realize the birds occurred in some areas. I've had birds that we've tagged that have summered in areas that are not described as red tails occurring from that same BNA red tail distribution map. That's awesome. That's where we want to be. We want to be pushing our understanding forward and refining that. So there will be changes, and I think that can be said for many other species as well. The other thing I want to say, particularly for the red-tailed hawk, is for the red-tailed hawk as an example of what isn't shown for many species. There's a unfortunate lack of population level distribution descriptions for many species. I was happy there, just based on what I understood about the species. That's the one that's featured on the website and that I've shown here in my talk. I've tried to outline where certain subspecies occur. And what I also tried to do is show areas where two subspecies came into contact and overlapped. That overlap is super important for understanding red-tailed ecology, evolution, and also identification. In most cases, those overlap zones are super broad. And so you have tons of intermixing of these birds with plumage characters, causing those intergrades or those hybrids, however you want to call them, to be super hard to identify to one population or the other. The more subspecies maps show that and describe that, I think the better our ability to be a little more conservative with our IDs will be. We'll understand the system a lot better, because that's super common at the subspecies level to have these overlap zones. So it's something that I want red-tailed hawk subspecies distribution map, but I think it can be done for a lot of raptors in general.
Great. Well, thank you so much, Bryce, for your helping us better understand and appreciate the species. I'll turn it over to Julie now for some final comments.
Okay, yeah, thank you so much, Bryce. That was really a fascinating look into just the incredible diversity of that species. That was wonderful. If you did have another question or two, you can reach out to Bryce. His email is in that last slide. But since we're past 1 o'clock, we should probably wrap it up. Next month, we're going to be talking about climate change. We're going to be hearing from Will Weber, who's the HMANA Climate Change or Climate Committee Chair. And that is happening on Wednesday, July 24th. So it's a ways off. You can register for that on our Facebook page, on our website now, or you can wait for my, there'll be a link in an e-newsletter that's going out soon. In other news, Raptorathon season for HMANA is winding down. Teams have completed their events, but there's still time to support them with a donation for another week or so. You could do that at HMANA.org. And registration for our fall conference is going live very soon. It's not quite ready. We're just working out the last few details. So stay tuned for that. But thanks to Bryce and to all of you for tuning in today, supporting Lunch and Learn and the work that HMANA does. If you value Raptor education programs like this one, we encourage you to become a HMANA member. It's really the best way to support Raptors, to show your support for Raptor monitoring, the education and conservation that HMANA does. So thanks again, everybody, and I hope to see you next month. Thanks again, Bryce. Have a good day, everyone.